Shruti Iyer from genetics will talk about solving genetic puzzles to understand cancer.
Why can't we cure cancer? Well, we can't cure something if we don't fully understand it. And we can't understand something if we're not using the right tools to study it. DNA sequencing is the tool used by geneticists to look at someone's DNA and try and find the faults in that DNA to see how it relates to the emergence of cancer. Now, there are two main platforms for DNA sequencing. Let's call them short and long. The short approach has been there for decades, and it involves breaking down your DNA into really small pieces, looking at the faults in each of these pieces, and trying to put the puzzle together to see how these faults relate with one another in causing cancer. Now, what if the faults you're looking at are much larger than each puzzle piece? What if they're spanned across multiple puzzle pieces? Well, now it's like trying to put together a 1,000-piece puzzle that has a lot of mistakes and no one's given you the box. Not easy, right? Well, it's this problem that long approaches try to circumvent. The long approach was developed just a few years ago, and the main goal was to break down your DNA into much larger pieces, giving you a clearer understanding of what each piece, as well as each fault in that piece, looks like from the get-go. But because this is new, the long approach has major drawbacks, like it's very expensive, and it's not as flexible to be able to study multiple patients at a time, which is what our goal is. And that's where my specific research comes into play. What I do is develop strategies to make these long approaches faster, cheaper, and more efficient so that we can look at thousands of patients at a time. One of the ways I do this is by narrowing our focus to only those parts of the puzzle that we really care about. Say you just want to look at the American flag. So scientists have found that there are specific parts of the DNA that closely relate to the occurrence of cancer. If you're looking at the flag and you want to look at the stars and the stripes, see if they're in the right place or not, etc., these are the kind of things that we're interested in. So do we really want to waste our time in trying to solve the whole puzzle? No. So what I've done is I've used specific chemicals that work like scissors and label makers. The scissors go and cut around the parts we want, making our unique special puzzle pieces. The label makers go and label all the parts we don't want, allowing us to pull them out from the reaction. This now gives us more time to be able to solve these tiny pieces across multiple patients, giving us a clearer idea of the faults across these patients and being able to connect that to cancer. So essentially, I'm helping solve cancer one puzzle piece at a time. Thank you.